When classifying and comparing drugs, it is important to look at their chemical structures to see how that determines their drug action. The three examples that you will see today are stereoisomerism, the ring strain of the beta-lactam ring, and solubility and uptake. Along with examining the structure, we will examine how these drugs interact with the body. As you know, there are two types of stereoisomerism, geometric and optical. These are both significant in drug action. First, we will look at how geometric isomerism plays a role in the effects of cisplatin. Many anti-cancer drugs, such as cisplatin, work by disrupting the function of DNA in cancer cells and preventing cell division from continuing. Cisplatin can be found as a cis isomer, cisplatin, or a trans isomer, transplatin. Cisplatin has the amines on the same side and the chlorides on the same side. In transplatin, these functional groups are on opposite sides. However, only cisplatin can be used for chemotherapy. When the drug goes into the body, the neutral complex exchanges one or both of its chloride groups for neutral water ligands, forming positively charged species. These positive species can bond to the negative charge found on DNA by exchanging the water ligands for bonds between platinum and nitrogen in adjacent molecules of the base guanine in the DNA. This modified strand of DNA will no longer have an effective repair process and will eventually lead to cell death. If we tried to create the process with the transplatin, the water ligands would be on opposite sides, and when it attempts to bond with the DNA strand, the orientation would simply not allow it to bond to adjacent guanine molecules. The other type of stereoisomerism, optical, presents itself in the two different enantiomers of thalidomide. Although enantiomers may have identical chemical properties, they can react differently when in a chiral environment. This is a very significant consideration because over 2/3 of drugs on the market are chiral. Drugs synthesized within cells naturally only have one enantiomer. However, drugs synthesized synthetically are known as racemates and contain a mixture of enantiomers. This creates an extra importance for drugs to be analyzed as to whether they are safe as racemates. This was not really considered until the thalidomide tragedy. Thalidomide was sold as a racemic mixture, and it was discovered later that the R and S isomers had completely different effects. R-thalidomide had the intended action, which was to induce sleep in pregnant women. However, S-thalidomide was teratogenic, which means that it was able to cross the placenta and produce serious deformities in the fetus. There is currently no regulatory mandate to develop drugs as strictly one enantiomer. However, it is becoming more and more common. One drug that you all know about that is marketed as a racemate is ibuprofen.
Beta-lactam ring action of penicillin. The structure of penicillin contains a nucleus of a 5-membered ring containing a sulfur atom known as a thiazolidine, attached to a 4-membered ring containing a cyclic amide group called the beta-lactam. The bond angles in this ring are reduced to about 90 degrees. Since they have sp2 and sp3 hybridized atomic orbitals, the atoms in the ring seek to form bonds with angles of 120 and 109.5 degrees. As a result, this puts strain onto the bonds and eventually weakens them. As a result, the ring breaks easily. The action of these beta-lactam antibiotics is to disrupt the formation of cell walls of bacteria by inhibiting a key bacterial enzyme, transpeptidase. As the drug approaches the enzyme, the high reactivity of the amide group in the ring causes it to irreversibly bind near the active site of the enzyme as the ring breaks. Inactivation of the enzyme in this way blocks the process of the cell wall construction within bacteria because it prevents polypeptide crosslinks forming between the mucopeptide chains. Without the strengthening links, the cell wall is then unable to support the bacterium, which then bursts and dies. However, some people are allergic to penicillin, so the drug is not for everyone to use.
Solubility and uptake. One thing that needs to be considered when looking at the action of a drug is how efficiently it can move throughout the body to reach its receptors. Most drugs that are soluble can be transported relatively easily throughout the body, which is an aqueous environment. However, if the drug is meant to reach the central nervous system, then the drug needs to reach the brain cells. But the human brain is surrounded by a membrane-bound structure known as the blood-brain barrier. This barrier protects the brain by restricting which chemicals can enter from the blood. It is hydrophobic and nonpolar, so for a drug to be able to penetrate this barrier, it must be more nonpolar, but still polar enough to be soluble to travel through the blood. An example of this is the difference between heroin and morphine. Both drugs are derived from opium, but morphine cannot cross the blood-brain barrier because it has two polar hydroxide groups. Heroin, on the other hand, has two ester groups rather than hydroxide, which is much less polar, so it is able to cross the barrier much easier, causing heroin to be much more potent than morphine because it reaches the brain faster and in higher concentrations. In conclusion, these three examples show how the structure of a molecule can affect the drug action and behavior inside the body.